Go.
Good afternoon, everyone. Today I'd like to share with you my vision for a genetically encoded and green and sustainable industrial future. And the way we're going to do that is by engineering molecular scaffolds. Molecular scaffolds are chemistry's version of circuit boards, where you would basically take modules that encode a particular function and assemble them in close proximity to generate a much more complex function. And scaffolds are ubiquitous in biology. Here are two examples for scaffolds that we find in biology. The left, typical scaffold that manufactures natural products. It turns out about 30% of our drugs, antibiotics, antifungals, anti-cancer drugs are manufactured by large protein scaffolds. On the other side, we've got cellulosomes, which are nature's way to break down complex material like lignocellulose into the individual sugars. Here, the bacterial cell would secrete the molecular scaffold to which enzymes are going to assemble the cellulases to break down the complex material. To give you a notion of what it is that I am going to, I don't know if I'll be able to play this, click here, so pause it. To give you a notion of what type of scaffolds I'd like to engineer, we're going to take our cues from long non-coding RNAs and see if I can do this.
אנחנו נדבר על זה כי זה in the real time it's going to be a big problem.
לא, אני יודע, זה אמור לגן אוטומטית על PC. That's just,
okay.
So give me that. So here we're going to see a DNA molecule. To the DNA molecule, we're going to attach an RNA molecule via something called triple helix interaction. Then we're going to also encode binding sites for RNA binding proteins. And to that, we're going to fuse your functional enzyme. And once we have one complex, we can then attach multiple such complexes that will enable to make basically would be a nano assembly line, a nano factory, or chemical circuit board. This comes at the heels of previous work where people have manufactured two-dimensional nucleic acid-based tiles to which they attach one or two enzymes, or individual three-dimensional structures. With slncRNAs, what we're going to be able to do is to marry those two worlds and be able to engineer much more complex functions that would be possible with either of these separately. The way we're going to go about doing that is by essentially developing the technology to figure out the rules how to build these objects. So what I'm going to do, what we're going to do is we're going to divide the aims and use language that we developed in the FET project, MRG grammar, using linguistic skills. First of all, we're going to divide our RNA into modules, functional modules. We call those words. Words in this case would be a DNA binding element, the RNA binding element, the protein elements, etc. Sentences would be combining these individual words into functional segments. Here we're going to increase the complexity. Finally, when we have sufficient amount of functional sentences, we're going to be able to hopefully extract the rules and develop an algorithm that would allow us to design a synthetic long non-coding RNA molecule. In a nutshell, this is what we do. Let's say we have a bunch of characterized words. We're going to then start putting together sentences. And eventually, we're going to get a sentence that actually makes sense. And based on that sentence, we're going to be able to start developing our rules database. Examples of the type of words. Here we're seeing a binding assay that we've been developing in my lab in recent years, where we can characterize RNA binding proteins and how they bind to RNA by installing the hairpin structure in the N-terminus of genes. This allows us to characterize multiple proteins, multiple binding sites, all in a high throughput fashion. One of the more intriguing elements of this research from a basic science perspective is the triple helix interaction. This element has the potential to not only make really cool synthetic long non-coding RNAs, but also can affect many fields in biology, therapeutics, anything you want. It is based on a different type of base pairing that was already discovered back in the 1950s called next to Watson-Crick base pairing. That's called Hookestein base pairing. It's another way of forming hydrogen bonds. However, unlike Watson-Crick, we do not really know the code for triple helix interactions. The problem within deciphering this code is the fact that on the one hand, we're going to need to have many targets, and on the other hand, we need to have many targets for the triple helix interactions and many candidates to those targets. So in order to on the one hand, solve this problem while controlling the complexity, this is why we chose to work with mammalian cells. Mammalian cells have 5 gigabases of DNA. That means that we probably have anywhere between hundreds to thousands triplex target sites. The second element of deciphering this code is to manufacture short oligos, where at particular positions, at bases, we're going to use mixed bases. Where, for example, instead of a C, we're going to have 30% C, 30% A, and 40% G. We've started developing this, and this is preliminary results for this type of interaction, where we've ordered these mixed oligos. And what you expect when you get an enrichment of such triplex-forming oligos in our assay is to get sequences back from sequencing that are enriched for Gs. The capital letters here are the barcode for the particular library, so we can sequence each one of these side libraries, and then we can increase the throughput of this by ordering many such libraries with different barcodes, thus characterizing the triple helix interaction. Once we move on to sentences, we're going to rely on another DNA synthesis technology to control complexity. Here we're going to order massive libraries of, again, single-stranded DNA oligos that are going to code multiple words. Here we're going to talk about between 10,000 and 50,000. In the example here, it's only 10,000, but what's going to be interesting, and this is the key to the entire proposal, is to be able to manufacture many of these at the same time. This allows us to characterize the important ones in the example here at the top and at the bottom, which we can then move on and take them to additional, more focused assays, such as imaging, single molecule and single cell level, where in this case we're going to replace the functional element by elements that encode for GFP, so we can actually see the molecules, or use different assays such as ShapeSeq, which allows us to get a structural footprint of these molecules in vivo and connect the expression and the functional element to the actual structure of the molecule. Finally, once we have enough rules, my lab has developed an extensive experience in numerical simulations, thermodynamic modeling, and kinetic modeling of biomolecular systems. This is an example of work we've done in recent years on modeling bacterial enhancers. It's going to look something like this, although for RNA it's going to be a different type of model, but using a similar approach. And a little bit about myself. I did my PhD at the Weizmann Institute in single molecule biophysics. I then transitioned to Caltech to synthetic biology. I've been PI at the Technion for the last 6 years. Grants, papers that we've published. This is my group. It's an interdisciplinary group involving having biologists, physicists, biotechnologists, bioinformaticians. We have post-PhD members of the group, graduate students, and most of these people have been involved in several papers in recent years. And to summarize,